Ribosomes are the cell organelles where translation process takes place. The ribosomes are made up of rRNA, that is ribosomal RNA, and proteins. They are present in the cell in large amount. They are present in both eukaryotic and prokaryotic cells. When we see the structure of ribosome, there are two subunits. One is large subunit and the other one is small subunit. Between prokaryote and eukaryote, there are certain differences in the structure of large and small subunit. In eukaryote, large subunit is 60S and small subunit is 40S. Okay. Generally, we call eukaryotic ribosomes are 80S ribosomes. Whereas when you come to prokaryote, their ribosome is 70S ribosome, and they have a 50S large subunit and 30S small subunit. S means Swedberg's unit. Actually, this unit is based on the sedimentation of ribosome when you do centrifugation techniques. Ribosomes are composed of two different sites: P site, A site, and in some textbook it is mentioned E site. Generally, these two sites are important, P and A site. A site is the acceptor site. Actually, in the process of translation, that is protein synthesis, between large subunit and small subunit, you can see mRNA get attached. This ribosomal complex is assumed to have three different sites. One site is A site, to which actually tRNA is bringing amino acids. Another site is P site, where protein synthesis takes place. Here actually peptide bond is formed between two amino acids. The amino acid present on A site and the amino acid present on P site. Mechanism of translation. In the translation, the first step is initiation, where you can see this is small ribosomal subunit. This is a modified mRNA. The ribosome, tRNA, mRNA, they all have to come together. For this, they require various proteins which are known as initiation factors. Several initiation factors are required for forming proper complex of tRNA, small subunit, and mRNA. Large subunit is also there, which will be added later on. At first, a complex of small ribosomal subunit and initiator tRNA, they will bind to 5' end of the mRNA. This complex will move towards 3'. When the starting codon, actually in every translation the starting codon is AUG. This represents methionine. When the AUG reaches at the P site, this is known as starting codon, the tRNA can easily form bond with AUG because they have UAC, which is an anticodon. You can see the codon anticodon binding here. Now the large subunit will also come and bind to the initiation complex with the help of a GTP. GTP is nothing but an energy provider. You can see a new tRNA can be now added to the A site. E site, P site, A site, three sites. A new tRNA can added to A site because there will be a codon here. Now many tRNA will come to this site, and only that tRNA which is carrying the proper anticodon to this codon can only bind to this site. In the elongation, that is next step, we can say the next step as elongation. In the elongation, the alignment of AUG codon with the anticodon of the initiated tRNA at P site the binding of second amino acyl tRNA, you can see next amino acyl tRNA when it binds, so requires the hydrolysis of again one molecule of GTP. Again energy is required. That means at every step it requires energy. Peptide bond formation occurs. Actually the bond happens between amino acid present on the P site with amino acid present on the A site. In between this peptide bond happens. That too, the carboxyl group of amino acid bound to the tRNA of P site and amino group of amino acid bound at the A site. इसके अमीनो ग्रुप इसके OH ग्रुप के बीच में पेप्टाइड बॉन्ड बनते. After forming the peptide bond, the entire chain will be shifted to tRNA present on the A site. At the same time, you can see the movement of entire complex towards 1,3' site. So what will happen? This tRNA will come to the E site and this tRNA will come to the P site. A site will be again empty. Again no tRNA can come to A site. Next codon will be occurring at the A site. This process continues and this is known as elongation, again with the help of elongation factors. Next is termination. You know there are various codons which does not represent for any amino acid. They are known as stop codons. The ribosome complex is moving from 5' to 3'. Here it is different. This side 5' to 3'. You can see new amino acids, new tRNA is accepted here, and the after formation tRNA is released through E site. This is A site, this is P site, and here it is A site. So this happening continuously. When it reaches a terminator, suppose this is terminator, that means at the end of the mRNA. You can see sometimes stop codons are present at the end, and when stop codon comes at the A site, see no tRNA will attach here because there is stop codon. Stop codon doesn't code for any amino acid. So when a stop codon reaches at A site, no tRNA will come and attach to that end. Thus comes a factor known as release factor. Then attaches a factor, that is release factor. This release factor actually releases large subunit, small subunit, mRNA, protein, tRNA. They will be removed separately and thus the process ends. So we will have a quick recap. In the process of initiation, first we can see the larger subunit, smaller subunit and tRNA at P site. A site is now ready to accept next tRNA. This process is known as initiation. Requires various factors, initiation factors. Now in the process of elongation, new tRNA will be added to the A site, and you can see a bond formation between the amino acid on the tRNAs, and the entire chain will be shifted to this one, new tRNA. The ribosome moves further, then the whole tRNA will reaches here. Again the A site will be empty. This continues, again requires help of various elongation factors. When the translation complex reaches at stop codon, no tRNA will bind here because no tRNA representing or can bind to stop codon. Thus release factor bind there. After the binding of release factor, protein, larger subunit, smaller subunit, mRNA all get separated. This is known as termination. I hope it is clear. Thank you.